What are the key findings from the recent H5N1 clinical trial, and how could this impact the future of intranasal vaccines compared to traditional methods?
We're actually not in a position to report the findings yet. We can report that we've been able to expand the data set that we're going to be presenting. When I was at the World Vaccine Congress, we were excited to present that. I think, especially for a phase 1 clinical trial, you always wonder how strong are the immunogenicity data going to be, not just the results, but the breadth of the results. And we're very excited to report that through some additional funding that we received as the trial went on, that we're going to report a really broad data set around not simply mucosal antibodies, but systemic responses, cellular responses, ADCC or antibody-dependent cell cytotoxicity responses. So we're quite excited about the breadth of data, and we're really looking forward to being able to make it public. We're not quite there yet. We need a few more weeks.
Are you able to give us a little bit of layout, maybe the methods, how you're approaching this trial?
In terms of the clinical study design, what we did was obviously with avian flu, with H5N1, you've got a naive set of patients, meaning they haven't been exposed to that virus in the past, in contrast to, say, a seasonal flu trial where we've all got background immunity from either previous vaccination or exposure. So what we did here was we set up the trial with 2 intranasal doses of our vaccine, our adjuvanted recombinant protein vaccine. And then later we came in with an intramuscular third dose. And the reason I'm avoiding using the term boost here is really we're using that intramuscular dose of vaccine, which is the Sanofi licensed H5 vaccine that's available today, really as a proxy for infection. Obviously, this isn't a challenge study, but with mucosal vaccines in general, we always know that the immune response is going to be different than how you judge an intramuscular vaccine, HAI antibodies, for example, in the case of flu. So what we really wanted to do is have the opportunity to prime these naive subjects with 2 doses of intranasal and then use an intramuscular boost to draw out that immune response, really to see whether what we've done is set the body up to respond to that exposure in a way that if that exposure was, say, a virus out in the real world, would be very robust and protective. So it looks like a 2 intranasal dose followed by an intramuscular boost, but that's not really how it's set up. It's really set up to have that intramuscular be a proxy for infection.
And can you tell us how the NanoVax platform addresses public concerns regarding vaccine safety and efficacy amid rising vaccine hesitancy?
Let me start with the last little bit of that phrase first, and it's probably the easiest one to answer. Obviously, some portion of vaccine hesitancy is needle phobia. Not all of it. People have a lot of reasons for being hesitant about vaccines, whether it's misinformation they have or previous experiences. I'm a family doctor by training, actually, a clinician for almost 30 years. And I can tell you that a lot of people just hate the idea of getting a needle stuck in their arm. And obviously, any intranasal vaccine, ours or others, gets you around that. So I think the vaccine hesitancy thing is very, very real as an issue at the conference. And at all of these conferences, I think we're all very aware that you can develop the best vaccines in the world, but it's incredibly obvious that that doesn't really matter if people won't take them. And we're seeing that now with the mRNA vaccines. I think the most recent studies show that only 10% of eligible Americans have gotten all of the series of recommended doses. It's very low. It's fantastic technology. It's protected a lot of people, but if people won't adopt it, whether it's because of information they've heard or reactions that they've had, feeling miserable for 24 hours afterwards, I've been getting my series, and I'll tell you, I'm a little tired of every 6 months having to go get an mRNA vaccine and then lie on the couch and be feverish for a day. So I think on the hesitancy side, any intranasal gets it around it. Changing to the first part of your question, safety. Everybody's always worried and should be appropriately worried about safety in any vaccine platform. I won't go too deep in this because it can get a little long, but the history of intranasal vaccination is interesting. We've known in intramuscular vaccines for a long, long time that adjuvants really matter, that the stimulator of the immune response, the adjuvant portion of a vaccine, separate from, say, the antigen, which is specific to the type of disease you're protecting against, that stimulator is really important for the breadth of protection, how well that vaccine will perform in keeping people from getting sick. We know that from intramuscular. Adjuvants really haven't been on the table for intranasal vaccination for quite a while because of safety concerns that arose from a single adjuvant system 20 years ago. Brna launched an influenza vaccine and post-market found out that the vaccine was causing Bell's palsy, a neurologic condition of the face. And I think the industry in general, appropriately, was so shocked by that. It got all the way through development, all the way into licensure, that really people said, let's stay away from adjuvants in the nose. let's just stay away from them for safety reasons. We think our platform addresses that very directly. The size of we have an oil and water emulsion, prevents passage into the neurologic system, anything that would cause Bell's palsy, the olfactory bulb in the nose. But we also don't just have this as a theory, we have it as evidence. We're now completing our third clinical trial. We've never seen a serious adverse event. And while that's hundreds, not tens of thousands of patients, the safety signals are very, very strong from these clinical trials. And more importantly, as I mentioned very briefly, there's a whole talk about it that I'll avoid. We have mechanism of action that is so separate from what Berna's vaccine experienced that we think our technology opens up a real opportunity to use adjuvants intranasally the way they have always been so important intramuscularly. It's an alternative to what most intranasal vaccines are, which are live attenuated viruses. That's the FluMist story, the only licensed vaccine out there. And most intranasal vaccines that are in development, whether it's for COVID or influenza or others, use a live attenuated virus approach. Live attenuated viruses have their own issues, have their own negatives and positives. No platform is perfect, but we think our platform technology opens up a realm that's otherwise closed or has been closed for quite a while. In terms of efficacy, really important that we remember we don't get infected through the arm to respiratory diseases. We get infected by breathing these diseases in, certainly in the case of influenza. So if we just start with first principles, if you get infected that way, an ideal vaccine approach is to mimic that infection as closely as you can without actually obviously causing the patient to be sick. But in terms of the immune response, it's just intuitive that if you can use an intranasal vaccine to vaccinate against disease that's transmitted intranasally, you're probably 3 steps ahead of putting something in an arm and hoping the immune system responds in the robust way that it would need to in order to prevent infection. One last point, what are we protecting from? We use that word loosely. Protection means many things, can mean many things. Typically, the way we've used it, certainly in the COVID era, we use it in terms of keeping somebody from getting very sick. What we've learned from COVID, or been reminded from COVID, is that that's half the story. The other half is keeping them from being contagious. There's scientific words for this: shedding, carriage. Point is having virus in your nose. You can be carrying virus around and making people around you sick, whether they be loved ones who are at high risk or young infants or just people in the general population. So we really want to expand what we mean when we say to protect. And I think intranasal vaccination, certainly our NanoVax platform, does that. It gets at not only disease, but contagion. And for these respiratory diseases, that's just really critical.